Hey, Ice, can we start? Because I see we have some good number.
Yeah, looks like the people in the waiting room has joined already. Looking good. The recording is started, and now Kato will introduce Professor Kate Adamala from University of Minnesota. Take it away, Kato.
Hey, thank you so much, Ice, for the moderation. Today, Ice will be our moderator, and I would like to welcome everyone who has joined it. Today's event, SynBio4ALL Africa guest speaker event 2. Today, we are very glad that we are hosting Professor Kate Adamala. Professor Kate Adamala is a professor of genetics at University of Minnesota in the United States, and she will be discussing about synthetic biology and biosecurity. A brief biography about Professor Kate Adamala. Professor Kate Adamala is a professor of genetics at University of Minnesota. Kate Adamala is an American synthetic biologist and a professor of genetics at University of Minnesota. Adamala's work includes contributions to the field of astrobiology, synthetic cell engineering, and biocomputing. Her research aims at understanding chemical principles of biology using artificial cells to create new tools for bioengineering, drug development, and foundational research. Her research interests span questions from the origin and earliest evolution of life using synthetic biology to colonize space to the future of biotechnology and medicine. Adamala's research on prebiotic RNA replication provided an experimental scenario for the RNA world hypothesis for the origin of life. She has worked on constructing liposome bioreactor synthetic cells. Kate is a founder and steering group member of the Build a Cell Initiative, an international collaboration for creation of synthetic live cells. She is a co-founder of synthetic cell company, SynLife. Adamala and Zustak demonstrated that non-enzymatic RNA replication in primitive protocells is only possible in presence of weak cation chelator like stearic acid, providing further evidence for central role of stearic acid as primordial metabolite. Also, Kate has worked on biosafety and biosecurity implications of emerging synthetic biology technologies, including horizon scanning and safeguard analysis. Her work particularly focuses on synthetic cell engineering and genetic code reprogramming, investigating implications of reprogramming living organisms beyond the bounds of natural biology. Dear friends and colleagues, today we have a privilege of having the incredible Professor Kate. Professor Kate, your presence adds a special touch to our gathering, and we are delighted to have you here with us today. Please, Ice, you can take on.
Thank you. Thank you so much for the great introduction. I will try to live up to half of that. So thanks everyone for being here. I will share my screen, and I would like, if anyone has any questions during the talk, please ask. I will make sure that I have my chat window open. So if anyone wants to type a question in chat or unmute yourself and just ask, please do. I think it's always more fun when talks like that are more of a conversation. If you can ask questions and I can answer, we can learn from each other rather than me just being a talking head on the screen. So if anyone has any questions at any point, please just stop me. And okay, so getting started. I promised to talk about engineering synthetic cells, and a lot of you are probably wondering, why is that something that anyone would be even interested in? What's the point of doing it? And that's a pretty good point, is why would we even bother making synthetic cells? What's the point of the field of synthetic cell engineering existing if you have this incredible diversity of life? This is just a tree of life that I got from some book. But on the biochemical level, when you look at the tree of life, it's not that different. On this incredible variety of life that we have on Earth right now, there is only one biochemistry. And within that one biochemistry, there is only one architecture for a cell. That's where we are right now as biologists. Despite this incredible variability of biology, the sample size for all of biology equals 1, because we only have one type of modern terrestrial life that's represented by one type of cell architecture. All the cells that we know of right now, they have the same type of DNA. They all use the same 22 amino acids. They all use the same 5 nucleobases for their DNA and RNA. The central dogma of biology is conserved across all domains of life. So from the chemical point of view, it's all the same. And that is the biggest question of biology, is how does that chemistry work? And how can we actually understand it? How can we understand how a cell works? And all of you are here under the brand of synthetic biology for all. And the premise of synthetic biology is that we can engineer biology. We can build things with biology that nature hasn't done before. And that's kind of a difficult if you don't truly understand a cell, if you don't know how a cell works. So that's the goal of our field, is we are trying to understand how exactly a natural cell works. And we're doing that by trying to simplify a cell. So instead of looking at this very complex natural cell, what instead we're trying to do is we're trying to make it simpler. And that's basically the premise of our field, is instead of trying to understand how a complex modern biological cell works, we admit that we can't. We cannot understand how all components go together in a complex living cell. So instead, we're trying to simplify it. We're trying to make it so that it actually can be not just understood, but also engineered. That's the goal. And so how are we getting there? Since we're bioengineers, we have 2 different options that we can choose. We can either study the complex life cells to death. We can study every single component of it, hoping that one day we'll be able to understand where everything goes. Or we can use in vitro systems, which are very few well-defined components that we know exactly where each of them goes. But the downside of this is that it doesn't have the complexity of a cell. You can study enzymatic reactions, you can study single tube enzymatic reactions as much as you want, but that will not really tell you much about the complexity of an actual living cell. So the third option, and this is what most of our field is doing, is let's try to build a cell. Let's try to build what we call a synthetic cell from scratch. And by doing that, we can maybe understand how a complex cell works. So instead of trying to understand this black box of a live natural cell, or instead of trying to work with single small molecules, we're building something that looks like a cell, but is simpler, much simpler than a complex natural cell. And there's obviously downsides of both of those approaches. If you're working with live cells and the synthetic cells made from complex living cells, you don't really understand exactly.
25 people.
Sorry, was that a question? Okay, if you have a question, say that again or type in chat. Okay, so 2 different approaches. We can make complex live cells that are simpler, or we can make the in vitro systems that are more complicated and start resembling a live cell. And so this is where the field of synthetic cell engineering is right now. We have those 2 approaches, the so-called bottom-up or top-down. And from that, we're trying to build a happy middle ground, a synthetic cell that's simple, understandable, and fully controllable. That's really the key, is we want something that's fully controllable. We want something that we can actually say we're engineering, rather than just poking with a stick like a natural cell and not being able to understand exactly how it works. So what is it good for? Well, the bottom-up approach produces cells that are very well defined. We know exactly what we're working with, but they're still dead as a doornail. They're not yet living. The top-down approach did produce a so-called minimal cell, the Craig-Venter mycoplasma-based minimal cell, but the problem is they're not completely defined. We don't know exactly where every molecule goes. And so this is the premise of our field, is we want to make something that's very well defined, that's a synthetic cell that's completely understood and engineerable. And so note that I still haven't told you what actually is a synthetic cell. So it's a really difficult question because there is no good definition of what a synthetic cell is because we don't have a good definition of life. And when I give those talks, usually people ask me, what is the definition of life? And I'm happy to discuss that, and we can talk about that for a long time because it's not a simple question and not a simple answer either. But to me, a synthetic cell is something that came from living components that were derived from living cells, but has not been living before the experiment started. So basically making life from non-living components. And if you think about it, every single living organism that exists on Earth right now, from you to smallest bacteria, all of that came from unbroken line of life that's been around on Earth for 3.8 billion years. So you are descendant of that very first living cell that started on Earth. And that has not changed. There has not been a second origins event since life started on Earth. And that's kind of funny when you think about it, that all life comes from that one population of the original cells that started once, and life never extinguished. So we are all descendants of that very first cell. Now that poses a problem, because if there has never been another origins event that made it to today, we don't know how to make a cell without breaking that cycle. So how do you get through from those non-living components to a living cell? Well, the biggest thing that we want to do is something that biology doesn't normally do, and that is controllability. How do you control every single element of biology? How do you control things that natural cells do not normally control? So some of those things that natural cells don't normally control are things like the size of a cell. You cannot take a normal living cell and change the size by order of magnitude. Your cell is going to die. You cannot change the translation system completely. You cannot change things like tRNA, or you cannot transplant a completely different foreign genome. Sorry, was that a question? No? Okay. You cannot control.
Sorry.
Yeah, no worries. So you cannot control all those elements of a cell. What you can control, though, is you can build a synthetic cell and control those elements in a synthetic chassis. This is where we truly become engineers. We can really say we're building a cell from scratch to specs, to those very specific specs. So what really, again, I still haven't told you what is a synthetic cell. So what is a synthetic cell in my lab is that it's a liposomal bioreactor that can express proteins. And that means we have a membrane, we have a liposomal membrane, and inside that membrane, we have all the components that are necessary for transcription, translation, gene expression. And all of those components together allow us to be able to say, okay, we've recreated central dogma of biology. We have genome that gets translated to proteins, that those proteins have specific functions. So this is what a synthetic cell, according to my definition, is. Other labs might disagree. Different people doing research in this area have different, their own kind of a pet definitions. And they're all valid since none of us actually made a living cell from non-living components yet. So when I work with synthetic cells, the outside of the cell is almost always a lipid membrane. So phospholipid bilayer membrane is not very different from your own cell membrane. So this is what you have in your own cells: a polar head groups hydrophobic tails of a lipid and some cholesterol for membrane fluidity. Now, sometimes we use fatty acids, probiotically plausible underivatized fatty acids, but for most experiments, we use the well-behaved modern phospholipids. Sometimes we don't use membranes. Sometimes we use droplet microfluidic systems. But this is a video taken by my postdoc, Theodore. The microfluidic systems allow us for high throughput formation of a lot of identical cells. The downside of it is, though, if there's no membrane, you can't use it, you can't release it to the environment, you can't use it as a drug because there's no specific separation of a membrane. So most of the time, we're membrane people. The stuff that really is interesting, at least to me, is what goes inside the synthetic cell. And that is a cell-free protein expression system. It's literally guts of a cell that were ground up and purified to recreate the central dogma, to recreate a translation system in this very controlled, well-defined environment. So what those components usually are is that we get ribosomes, acyltransferases, tRNA from a source organism, most of the time bacteria, and then we add all the small molecules that are needed for transcription and translation. So we add energy in a form of ATP and energy regeneration systems. We add all the nucleotides, amino acids. We add some modified tRNA, and then we add genomes. And if you mix all of that together under specific conditions, out comes protein products. So this is kind of the basic anatomy of a synthetic cell. Now, what can we do with it? What is it good for? There is a lot of really cool applications that are both practically useful and appeal to people that are interested in foundational research. By building synthetic cells, we can understand natural biology better. We can understand how cells work, how natural cells work. We can also build tools for understanding biology. So all of you who've ever done any experimental synthetic biology or biological engineering, you're all probably familiar with Gibson ligation. It's the most common cloning method used these days. And Gibson ligation was actually developed by Dan Gibson when he was working at the Craig Venter Institute in the process of making their first mycoplasma-based synthetic cell. So that shows you that there's this interplay between the tools we build, because we have to build those tools to make synthetic cells, and then those tools become just useful general synthetic biology tools beyond the field of synthetic cell engineering. Gibson ligation is the best example of that, but there are other cloning methods, other mRNA imaging methods that were developed for our field first, and there's a ton of microfluidic methods that development of those methods has been driven by the synthetic cell engineering, but then they took off and they are now used in different fields. There's also biomedical applications, things like diagnostics and therapy. We can use synthetic cells to make better drugs, to make better possible drug candidates. We can use it for metabolic engineering. I think the last talk you had in this series was by Michael from LanzaTech. He probably talked a lot about the great potential of metabolic engineering. And a lot of the prototyping of metabolic engineering circuits these days is made in cell-free systems because they're quicker, faster to work with. Then there are the fun applications. There is applications in biocomputing. There's been a lot of work on engineering synthetic genetic circuits in vitro systems. There are applications for space exploration, for example, for making astropharmacy platforms, and I'll show you one example in a moment. Then there's applications in studying the origin of life, because building synthetic cells is like building the simplest earliest cells on Earth. And then finally, there is a lot of biosafety and biosecurity applications. We're literally building a new chassis. So in order to do that, we have to do it in a safe, secure way. Okay, so I just showed you a lot of cool sounding applications, and I claim that we can do all of that with cells that we built from scratch, with cells that we built from non-living components. So that sounds kind of crazy. I totally agree that this doesn't sound like something that's realistically happening. So before I show you the state of the art right now and how close we are to truly building a living cell from non-living components, I want to show you a quote from an article that was published over 100 years ago in the New York Times. So New York Times, a very respected publication, they published an essay on the feasibility of flight heavier than air. So remember, it's 1903. We have balloons. So we know that a human can build a machine that can fly, but balloons are lighter than air. That's how they fly. Now, people have been speculating that it's possible to build a machine heavier than air, flying machine heavier than air. And this article was published with a very reasonable, very concise argument that essentially say it's impossible. You might as well not bother. Don't bother building a flying machine heavier than air, because it's a waste of time and waste of resources. So this very respectable journal published an article, and then 2 months later, the Wright brothers flew their first airplane. And that just shows you that if you tell engineers and scientists that something is impossible, we're just going out and going to do it. We're not going to listen to people that say it's impossible, because if we believe it's possible, we will get it done. And this is one of my favorite examples. There's obviously been a lot of examples in history where people say something is scientifically impossible just to see it done sooner or later. And that's the lessons that our field is taking. People say that building a living organism from non-living components is impossible. And our answer to it is wait and see, because we're about to do it. So now I'm going to show you some unpublished data from my lab where we're demonstrating, this is basically state of the art in the field right now, how close we are to really making that a reality. So the hallmark of life is a cell cycle. Every known living cell has a cell cycle. And so what we did is we engineered a cell cycle in a synthetic cell, in a synthetic minimal cell system. So how we did it is we start with a membrane that encapsulates the whole thing. And inside that membrane, there is a genome replication system. That genome replication is done with a rolling circle polymerase called Phi29. Now, we also have protein expression. So protein expression is done with the so-called pure system. Someone's trying to draw on slides. If you want to emphasize something or ask a question, feel free. Okay, so we have gene expression that's done with the pure system. Then we have membrane pores. Those membrane pores are to let nutrients in and waste out. Then what we have is we have a feeding and growth system. The feeding and growth is allows us to basically help to grow the cell, add all the components that we can't make inside, and add more lipids. So basically to be able to add more lipids to the membrane.
Okay. Right.
Now, we also have evolution system. So we're evolving our cells so that we can basically observe things like natural selection, origin of Darwinian evolution. And we have division. Division here is mechanical, so we're doing division by extrusion. Last but not least, we also have a, let me find it. Okay, we also have a, oops, my slide changer stopped working. All right. Last but not least, we have a generation counter. The generation counter allows us to prove that in this whole cell cycle, there is a population of cells that goes through all the cycles of growth and replication. So all of that allows us to go through minimum of 5 cycles of that complete cell cycle of growth and replication. We can do more, but 5 is what we really have good data for. So right now we're claiming 5. We also can show competition. We can show competition between fast-growing and slow-growing cells. And we can show competition between fast feeding that leads to more offspring. So essentially what we shown is something that almost fulfills the NASA definition of life. So that definition is self-replicating, self-sustaining chemical system capable of Darwinian evolution. Our system is capable of Darwinian evolution because it grows and replicates. It is not self-sustaining. Now, I mentioned a few minutes earlier that there is a big debate about what really is the definition of life. So our system is not self-sustaining, which is why I think it's not living. But when you think about the NASA definition of life, no living system is truly self-sustaining. According to the NASA definition of life, neither of you is alive because none of you is self-sustaining. You cannot produce all the nutrients that you need for life by yourself. You need to eat like we all do. So it is that kind of emphasizes the trickiness of the definitions of life and how it's really difficult to figure out exactly what is life. And so it will be really difficult to figure out when are we done, we as a field of synthetic cell engineering. Okay, so this is the state of the art. This is the where are we really as synthetic cell engineers? And now I would like to show you a highlight of another project, a collaboration that I had a pleasure to be a part of with people from mission development and biotech and pharma companies. And what we did is we asked the question, can synthetic cell and cell-free systems be used for astropharmacy? So what is astropharmacy? It's an idea that you should be able to make drugs under spaceflight conditions on demand, on short notice from compounds that are shelf stable. This is the premise of the needs for long-term space missions. When we're going to send people to Mars or to the moon, we're going to have to be able to produce pharmaceuticals on demand, on short notice, without knowing in advance what is going to be needed during that long-term mission. But when you think about it, if you have that kind of a robust biomanufacturing platform, it's also going to be useful for things like on-demand biomanufacturing, field deployment under terrestrial conditions. So even though this is an astropharmacy project, it also has the benefits for production of pharmaceuticals, production of small molecules under low resource conditions on Earth. So what we did is we asked, can two cell-free reactions happen under spaceflight conditions? And those two reactions are a synthesis of a functional RNA molecule, RNA aptamer that induces fluorescence of a small molecule, and a production of a small fluorescent ligand peptide. So both of those, transcription, RNA folding, and protein expression, were done from a lyophilized sample. So samples that was freeze-dried and stored at room temperature. So that shows you this kind of a robustness and the ability to store those reagents at room temperature before use. So there is a video that I have to get up at 4 a.m. to watch it, and it's obviously super exciting for me because it's my own sample going into space. So I love showing that video to people because I grew up as a science fiction nerd, and being able to actually work on projects that do get launched was fantastic to me. So we did prepare that payload and we did launch it, and it was a sample return. So we did get it back, and we can say that yes, cell-free systems do work under spaceflight conditions. It's possible to make protein expression experiments under spaceflight conditions from a lyophilized sample. And so that shows you that there really is a promise in this technology. Some of those applications that we're talking about, like being able to make pharmaceuticals on demand, being able to make biologics on demand, doing all of those medical and biocomputing applications, these are actually possible because those systems are very robust. So this is just to remind you why the field is so excited about some of those applications. This is just an example of some of the things that we're looking at right now, some of the applications that we're looking at right now. And one thing that I want to show you that is especially related to the things that are of interest to all of you and really anyone who has any needs for diagnostics, therapy, and biological medicine, there is this graph from a review that was published a few years ago that shows you the relationship between investment in pharma R&D, so research and development in pharma companies, and the number of drugs that were brought to the market. And as you see, it's inversely proportional, which is kind of funny because it almost looks like the more money we're putting into pharma R&D, the less drugs, the less number of drugs we're actually getting. And so something's wrong, something's not right with this. And that might indicate that we are in a need of a new chassis. We're in a need of new type of tool or new type of a discovery chassis that would reverse that trend, that would allow us to actually make new drugs and make new applications more efficiently. And so that's the niche that our field is trying to fill, which is we say we have a new chassis, and it's still a very early field, but we do show some very promising biologicals and potential drug candidate results. So I'll show you the next few slides of results that are not from my lab, but from collaborators. So one of the interesting recent results is that synthetic cells injected into a live mouse can shrink tumors. So what you see here is stained breast cancer model tumor. And if you combine that with a synthetic cell that expresses a specific protein, those synthetic cells shrink the tumors, shrink the tumor models. And so they kill the tumor cells. Now, another application is in organoid growth. This is work from Avi Schroeder's lab at Technion. We can use synthetic cells injected into an organoid to grow vasculature. The yellow on those slides is stained vasculature. So you can see that it's possible to grow new vasculature in an organoid by using synthetic cells that secrete a specific growth hormone that induces that growth of vasculature. It's also possible to use cell-free systems. So these are the freeze-dried, the lyophilized cell-free systems that we used for that spaceflight experiments that I showed you. And these can be used to make vaccines that actually induce immune response in mammals. So what you see here is an example of work from Jim Collins's lab, where they took a freeze-dried lyophilized cell-free system, and they used it to make vaccine that then actually produced measurable antibodies. Yep.
Hey, Kate, we have a question in the chat.
All right. Oh, I see. Awesome. Can these synthetic cells become malignant too? How do you make sure that they can proliferate uncontrollably when induced in the body? That's a very good question. And the reason they cannot become malignant is because they do not self-replicate. That's the advantage that we have over the CAR T-cell therapies, is synthetic cells in this design, they do not have the mechanisms for self-replication. So you inject them into a patient, they go do what is that they need to do, and then they die, or I mean, they're already dead. They dissolve, they get metabolized. So they're more like a long-release therapeutic rather than an actual living cell that can be injected and then give you something like a blood cancer. And this has been used, that's why we're getting funding for this work from the same people that were developing CAR T-cell therapies, because our cells work similarly, but they don't have the ability to self-replicate. And I see a follow-up question: by which mechanism do they shrink the tumor cells? So there are at least two different mechanisms that have been demonstrated so far. In that slide that I showed you, that work from Avi Schroeder's lab, the shrinking of the breast cancer tumor was done by secreting a toxin. So the synthetic cells had a plasmid that encoded a membrane channel that was a toxin. And that channel was made and secreted, and then that killed the cancer cells by proximity. So that channel, it can migrate across adjacent membranes, and it basically created indiscriminate holes in a membrane of cancer cells. So it basically lysed them. Now, this is not very targeted. This only works if you can get your synthetic cell therapeutics right next to the tumor. So it works for solid tumors, but it wouldn't work for tumors that are small cells. So another mechanism that's been demonstrated very recently is that synthetic cells can be decorated with antibodies on a surface. And those antibodies can target to a cancer cell and then work very much like CAR T cells. So we can decorate synthetic cells with both an antibody against a specific marker on a tumor and a protein that will recruit your own immune system to that site. So in a way, they're kind of like flagging the cancer. This is the cancer, it's here, come immune system, come eat it. So these are the two mechanisms that have been demonstrated so far. And I know people have ideas for other mechanisms, but for now, these are the two that are the most used. Okay, if you have any follow-up questions, please ask, and I'll keep an eye on chat. Okay, another application that's been demonstrated recently was from Fritz Simmel's lab, and what they've shown is that you can make active bacteriophages in a cell-free system. So bacteriophages are viruses for bacteria. They're viruses that infect bacteria. And one of the biggest problems in modern medicine is antibiotic resistance. We're getting all those drugs that are all those antibiotics, and we're using them like there's no tomorrow. And so the bugs are getting resistance to it. There's more and more antibiotic resistance strains present in hospitals, in the field. So especially for hospitals with low resources, they have to use the antibiotics because there's no other way, but that just breeds those antibiotic resistant strains, and we don't have a good way of fighting that. So this is what this work demonstrates, is that it's possible to create a bacteriophage, which bacteria are not resistant to, because it's not a small molecule antibiotic, it's a phage, it's a virus that actually attacks the bacteria. And it can create this therapeutic phages on site and then kill bacteria. So this is one of the kind of applications that can have a direct consequences both for human health and for biotechnological applications if you want to, for example, keep your bioreactors clean. So I see a question. Isn't it possible for these synthetic cells to alter normal functioning of living body cells, especially when introduced in the body for therapeutic purposes? Yes, it's very possible. That's what we're hoping for, in fact. So they are stealth to the immune system because we designed them with membranes that are pegylated, and they do affect functioning in a way that we want. So, for example, one of the synthetic cell-based therapeutics that is in preclinical at the preclinical stage right now is synthetic cells that secrete an enzyme that a body is missing. And so those so-called enzyme replacement therapies is when you're introducing an enzyme to literally alter functioning of a body, and you're introducing that enzyme because the body's not making enough of that enzyme, or you're under conditions where you need that enzyme that your body's not making. And so that's one of the examples where synthetic cells are altering the functioning of a body in the way we want, obviously, in a good way. There are alternatives where if the products of the cells are toxic, they could create side effects like not dissimilar to chemotherapies. And that's something that every single formulation of synthetic cells has to deal with. So we basically have to test them for both the immunogenicity of the compounds themselves and also how do we affect that, what's the effect on the host. So this is something that for every formulation you have to do that separate. Okay, so these were some examples of applications that we we're working on and our collaborators are working on. And I want to spend the last couple minutes talking about not the science itself, but how do we make science? How do we do that science? How do we make those experiments happen? And so I'm a coordinator of this initiative called Build a Cell, which is an international community for people engineering synthetic cells. And this is a map with our synthetic cell member labs. And I would like to sadly note that there is no labs from Africa, from the whole continent. There's not a single lab that's involved in Build-A-Cell right now. So if you know anyone who's interested in any aspects of that work, we would love to have you. We would love to work with you. I see another question. Don't the synthetic cells induce immune reaction in the body? As far as we know, the answer is no. They do not, and that's because we use lipids that make them stealth to the immune system. There are ways in which you can do that when you add those peg brushes, long peg brushes on your lipids, kind of like people do with other liposome encapsulated drugs like Doxil, for example. So we hide our synthetic cell payload inside that peg shell, inside that lipid shell, that pegylated lipid shell, and that makes it invisible to the immune system. So no one has ever injected them into humans yet, but from animal studies, from mouse and rat studies, we can tell that there is no cytokine storm. There is no excessive immune response, as far as we can tell. So going back to our organizing, the Build-A-Cell community is an international open community, and our goal is to help engineering synthetic cells. So we do not define what life is. I mentioned earlier, it's a really difficult problem. We cannot define what life is. What we do instead is we're coordinating research effort, and we're doing that by in-person meetings. So we're meeting twice a year all over the country, and these are international meetings. We always have guests from other countries. Those workshops are every 6 months to bring people together to coordinate our outreach activities, public education activities, and we also work on science policy. We work on biosafety, biosecurity implications, and big shout out to NSF for funding us. And I wrapped up on time, which is unusual for me. I want to thank everyone in my lab and the funding agencies that support my lab. This has been an amazing adventure. I'm still a relatively junior PI. I've started my lab a few years ago, and I decided I want to make fake cells from scratch. And people actually joined my lab, and people gave me money to do it. And that's amazing to me. That's something that I'm really very grateful for. And I would love to work with more people, and I would like to be able to collaborate with more people on different applications. All right, so that's all I had to say. And if anyone has any more questions, I would love to have a discussion. Or if you have any questions that you don't want to ask in public, if you Google me, my email address is publicly available, so you can always ask me questions over email.
Thanks a lot, Kate. That's amazing. Yeah, so like for the audience, if you have any questions, feel free to type into the chat, or you can also unmute yourself and like speak up as well. I see, is that a hand raise? It might be a clapping. Oh yeah, okay. Adamala, if you have question, please like feel free to unmute yourself.
Okay, so could you please elaborate on using the your system for biologics? How the systems can work with biologics?
You mean how the systems can, is the question how do those systems can work with biological drugs?
Yes, yes.
Yeah, so they produce the biological drugs. They don't work with them by themselves. They produce biological drugs. So they're basically they produce a drug, secrete it, and then as they secrete it, the drug goes in and then does whatever it needs to do. So for example, if you're producing a biological drug against a cancer, then the synthetic cell makes the drug, secretes it, and then the drug goes and does whatever is needed. So the biological drugs don't interact directly with the synthetic cells. They're being made by synthetic cells and then move on to do whatever it is that they're paid to do.
Okay, how will the targeting be overcome? How do you target the synthetic cells to its location?
So we target right now, two ways of targeting has been shown. One is direct injection. So people have done intratumor injections. There is a lab at Technion run by Avi Schroeder who's pioneering this work. They have mice with breast cancer tumor models, and they directly inject into the tumor. So that's the physical targeting. And kind of a more sophisticated way of targeting is that we label the synthetic cell with antibodies, and then we inject them into the bloodstream, mostly in rat studies through the tail vein. And as they have those antibodies on the surface, they go into wherever the tumor is by binding to those markers on the cancer cell surface. So it's the kind of the same homing mechanism as CAR T cells.
Thank you. Yeah.
Thank you.
Thanks a lot for the question as well. Is there any other questions from the audience? Again, if like unmuting, or if you're in a space where it's hard to speak up, you can also type. Yeah, maybe like can I do a quick follow-up question, Kate, on that one? When the Bayesian asks about biologics, I think like there are some other like biological parts like vaccine or other parts that could be they could benefit from the synthetic cells platform, which could be distinct from those biologics or like vaccines made from living cells. So can you comment a little bit on that?
So definitely there are at least few groups that work on developing different kinds of short peptides, but either for vaccine purposes or for other therapeutic purposes. And those are made from unnatural amino acids. So that's something that you cannot do in living cells. We can translate many unnatural amino acids that we cannot translate through the normal, normal E. coli expression. And so that's kind of the main area that's being pursued right now is unnatural building blocks and anything you can do with those never-born biopolymers. And I see two questions in chat. How biosecurity can be linked to synthetic DNA? So the main way in which we're doing it right now is synthesis screening. When you order synthetic DNA, if you're ordering from a company that is member of that international organization of the gene synthesis companies, which most are or should be, they screen against a known list of those so-called select agents, so pathogens that people should not be working with. And that's the main kind of a line of defense is the computerized screening at this DNA synthesis stage. Then there are local safety security protocols. Every lab has or should have standard operating procedures for neutralizing synthetic DNA before you dispose of it. And so that is kind of another line of defense against accidental release into the environment. And I see another question. Can you highlight a bit on how these synthetic cells are regulated with respect to biosecurity? So unfortunately, right now, they are not regulated. And that's something that we are trying to change. We are trying to develop community-based, so we're basically trying to self-regulate ourselves. We're trying to develop guidelines for both safety and security. So SOPs, those operating procedures, protocols for disposal, rules for constructing different kinds or not constructing different kinds of cells, and also develop some sort of a mass analysis on what can go wrong with those technologies. This is all right now grassroots, community-driven by people who do work on those technologies in the lab. Because for now, the regulatory agencies have not paid attention, and it's changing slowly. At least in the US and Europe, we had good luck talking to the regulators and policymakers, and they're starting to realize that we exist and we probably should be regulated, but it's a slow process. Okay, another question.
Okay.
Oh,
thank you so much, Kate. I have it's in form of a question, but I'm asking how could Build a Cell collaborate with SynBio4ALL Africa to support the scientists across Africa, because scientists across Africa really, they need capacity building, they need support in different perspectives, and they also need to reach the technologies your lab and other labs have reached. How can we collaborate to support these scientists?
So the best way to, collaborate, there are two ways to collaborate. One is less effective, one is a bit easier, the other one is more effective but harder. The less effective but easier is to get your people over to our meetings. And we have some funding for that. So we could start talking and we could start basically collaborating in person on developing projects right away. But to do any projects, we need money. And that's the biggest thing, is do we need to fundraise as a community to build capacity? Because as you said, the biggest problem is that you don't have the infrastructure right now. You don't have the physical capacity. You have the personnel, you have motivated, talented people that know what they want to do, but they don't have resources to do that. And this is something that I think can be addressed by private foundations, because governments are not that interested. I'm mostly funded by US government. They're not going to fund a development of a lab in Africa because what's their interest? And logically so. I mean, they're funded by US taxpayers, so they have to support US initiatives. There are some joint initiatives. I collaborate with people in different countries through US government funding, but those initiatives are reciprocal. So for example, I have a collaboration with someone in the Netherlands right now, but I'm paid by my government and my Dutch collaborator is paid by their government. So the governments agreed on those joint collaborations. This is difficult because that requires your local government to put up money as well. And I realize that's very difficult. So I think the best way to do that would be to approach foundations that are interested in global development. So developing global infrastructure and developing communities and opportunities for communities, and make those people realize that bioeconomy is happening. And if people in Africa are not given the tools to develop your own bioeconomy capacity, you're going to be left behind. And that's something that we can't afford, because it's a huge continent with both talent and resources. And especially in bioeconomy, when you're talking about making things with biology, you're talking about making compounds with natural bioresources. And that's something that every country, most countries in Africa has a ton of it. You've got sun, you've got water, you've got soil. That means you are a perfect candidate to be a platform for bioeconomy. You have people that can take advantage of it. And what you need is infrastructure. And to develop that, there needs to be interest from private investors, because that cannot be done on the government level. There needs to be private investors that say, for example, I want to build a bioprocessing facility in Uganda. How do I do that? And then we can provide expertise, because that's what we know how to do. And you provide the human resources, because you have people on the ground that would like to be doing that. So that's obviously difficult, but that's the way to go forward, is to find people that are interested in generally developing resources and infrastructure in Africa, and convince those people that synthetic biology is what they should be investing in. That obviously, they're investing in healthcare, they're investing in infrastructure on the ground, in education, but these are all investments to, make up the kind of the basic resources. If you want to not just catch up, but be actually competitive, you have to invest in synthetic biology. That's the new economy. The 21st century is called the century of biology for a reason. We really are developing those technologies right now. And so you don't have to be a follower. You can actually be building those capacities on your own and be leading development of those capacities, but you need investment in resources. So that's my very long and rambling answer to your question, but that's where I see we should be going. Is it should not be like right now, where a handful of richest countries are leading the bioeconomy revolution. This should be something that everyone is doing because everyone has those resources. And that's something I feel really strongly about. And that's what we should be doing.
Thank you so much. But I think we shall continue a bigger collaboration with you and Build a Cell, see how best we can support all these scientists who are on the call, because everyone on this call is interested in whatever you have talked about, and they have the brains and they would like to put it into action also at the continental level of Africa. Yeah, we can continue with more questions.
Yep.
Thanks a lot,
All
Kate.
right, I see
I
see a couple more questions.
Yep.
Yeah, I see a couple more questions in chat. Okay, so the last one I didn't answer. Okay, the question is, how can we ensure that synthetic cells are biocompatible within a living organism and won't trigger harmful immune responses? So the biocompatibility is easy. They are made of the same compounds as natural cells. The proteins that are made in synthetic cells are biocompatible by design because they are made from the same building blocks. Now, the immune response is not triggered because we encapsulate them in those stealth vesicles, so vesicles that are covered on the surface with compounds that don't induce immune response. Okay, the next question is, when introducing synthetic cells into the body for corrective therapeutics, how do you ensure that the right dosage of cells is introduced so as to avoid the overproduction of the desired product in the body, for instance, enzymes? That's literally $1 million question. The first synthetic cell drugs that will hit the market will have to answer that question. And unfortunately, the only way to answer that question is clinical trials. We don't know in advance. We can do back of the envelope calculations, which we are doing right now. Knowing the volume of the blood and the concentration of the enzyme you need, you can sort of figure that out, but you will not know for sure until you actually inject the damn thing into a patient. And that's what clinical trials are for. We are going to know the dosage exactly once we have the patients that are taking it. Another question, how will you organize and package the genetic material within the cell when building synthetic cells? So the short answer is we don't. The synthetic cells right now are mostly modeled after prokaryotic cells. So our chromosome is just naked DNA. We don't have histones, we don't pack it, just like bacteria. Bacteria have those circular chromosomes that are not organized into much of the secondary structure. And for now, it's good enough. The genomes are small enough. By the time they get big enough to need packing, we'll face that problem when we get there.
Is there any more question? Feel free to unmute yourself as well, and feel free to type. I think we can like continue for a few more questions, and that would be like good for the time that we have. In the meantime, I have one question that originated from the meeting that I first meet you, Kate. So in our like roadmap about space, we talk about like how do we use cell-free system or synthetic cells or these platform in like some place that were very far away, that was similar to like how the space and habitat or space environment is. Can you like touch on that a bit? Like how do you feel, or how do you think like these technology based on cell-free system or synthetic cells can be applied in like some place where infrastructure is not that like well developed? For example, like in some African countries where they were just studying, like how can these help them like advance further?
Yes, that's one of the biggest appeals of that technology to me, because it allows us to bring those cutting edge biological drugs to the places that don't have as many resources. And so cell-free systems can be lyophilized and they can be stored at room temperature. So if you do that, you don't have to maintain a cold chain. I know that was a very big problem during the post-COVID vaccination campaign. Is it's difficult to deliver a vaccine that needs to be kept at -80 Celsius to rural places and to places without a cold chain. And so if you can make your biological drug or your vaccine from something that's stored at room temperature, that obviously lowers that barrier because you transport your substrates, your lyophilized cell-free system and your DNA with just a normal package. You can put it on the back of your truck and drive wherever you want. And then you mixed it with water on site and it produces, the reaction starts and it produces a correct product. So that's the that's the biggest advantage, is that it is a biomanufacturing platform that can be stored and transported at room temperature. That's obviously attractive for spaceflight because you don't need the atmos, because you can lyophilize the whole thing, but also you can just do it in any other place. You can do it in remote locations.
Thanks a lot. That's great. There are a few questions down there, but I have a very quick follow-up question about that. When you say to room temperature, can you elaborate more, a little bit more? If the room temperature is only below 30 degrees Celsius, or like in some places in Africa where it might be over 40, is that still okay?
So actually, I can answer that quantitatively right now. We did the tests, and it is possible to store them. So actually, before our flight, before that flight, it was sitting on a tarmac in Virginia in the summer for 2 days because we were waiting for a weather window. And so, it gets really hot in Virginia in the summer. I don't know what was the recorded temperature, but it was way over 100. So like probably over 40 Celsius. And it survived, it was fine. Now we did quantitative studies, and it survives up to at least 45 degrees Celsius. We haven't tested higher, but up to 45, we heated it up and then we reconstituted it, and it did lose some activity, but it was still active. So yes, room temperature anywhere, including Africa.
That sounds great. Yeah, we have 2 questions in the chat right now. I can help ask the question. Do you think that genomic ancestry could affect response to synthetic cells in individuals of various ancestry? Question from Tequila.
Absolutely, it could, and this is one of the areas where we don't have enough data. Because when you look at clinical trials, they're usually made on males of European ancestry. That's the best studied population in the world. If you happen to be not a male of European ancestry, you're screwed. You can hope that the therapies will work on you, but there is not that much data. And so that's something that we are really need is, that's why one of the reasons why I think we should be developing those local centers for bioeconomy is to get the local biotech and local biomedical research, because no one's going to be interested in testing it on your population unless you're there doing it. And that's, I think, is the best way to kind of ensure that we know exactly how ancestry affects activity of those tailored biologics. It's called personalized medicine. But again, it's personalized to whatever patient population you have receiving it. And right now, the patient populations receiving those drugs are, again, mostly in the countries that can afford it. And so that's, to directly answer the question, I think, yes, it could affect it. I don't know how, because we don't have the data right now.
Sounds great. I think the last question in the chat right now from Ofoiworth is, since the genome doesn't need packing with histones, aren't they exposed to environmental conditions that may alter the genes, consequently resulting to translation of undesirable products? Or how is it like protected or regulated?
So it's protected because it's inside the vesicle. So it's protected just like a bacterial genome is protected. It's inside a cytoplasm of a synthetic cell, so it's not exposed to the elements. It is inside the cell. It's just not packed as tightly as eukaryotic genome.
Perfect. Oh, there's just one more question coming in. Instead of waiting for clinical tests, can enzymes be used to control overdose in the lab?
So actually, the one enzyme replacement therapy of synthetic cells that is being investigated right now is actually for enzyme that helps with overdose patients. I mean, we have to wait for clinical tests. There is the community of do-it-yourself biolabs, the biohacker people that are injecting untested things into themselves. One of them already killed himself in the process of doing that. So I do not endorse. I'm frustrated with the slow pace of clinical trial progress too, but this is necessary. We are finding, there is a reason this project, this is such a slow and expensive process. A lot of the promising drugs go to die in clinical trials because of side effects. So we do need those trials. We cannot just take a promising enzyme replacement therapy or any drug and just go inject it into patients. That's not, unfortunately, as much as I would love to be able to shorten that path is there for a reason. That path is there for our safety.
Thanks a lot, Kate.
Thank
So
you.
if there's no more questions, or if there are more questions, we will be happy to, like I think Kate said, you guys can reach out to Kate like through online like channel as well, and we will have these like webinar recorded, and we will like make it publicly available. So if you have any question, you can also comment on the YouTube recording that we will have, and some of us will like get there and like maybe talk to you later, and we can provide some simple answers on that. There's one last question, which is great. Viewers asked, like, will you please recommend a site where I can access more information about today's discussion? So do you have any suggested site on that, Kate?
I can paste you two links. So one is our Build a Cell community. And this is a great place for all the Build a Cell activities, for all the kind of ways to connect with the community. And another one is we have just published a roadmap to engineering synthetic cells. It's kind of a big document that discusses all the science that I was talking about today, and the link is available in chat for that. So this is a very good starting point if you want to learn more about the applications and the science. So this is a research paper that kind of discusses all of those implications, both of the research and the practical applications.
Yeah, and if any of the audience here cannot access to like some of these because of paywall or anything, like feel free to contact us, the staff of SynBio4ALL, and we can probably help with that. I
can send PDF to anyone who wants it. Payloads are stupid, and as an author, I have a right to share my PDFs, so I will share it freely with anyone who wants it.
Okay, Kate. I would request Kate that you share that PDF with SynBio4ALL Africa. We have a website. We normally post there everything, whatever we have. It's free content, and everyone will be in position of accessing it at any time.
Fantastic.
Yep. So if there's no more question for the interest of time,
there is a question. There is a question from Brian.
Okay. Please.
Yeah, I have a question.
Yeah.
I'm asking that, being that you don't have any lab for the biology in Africa, and there's young Africans who are very interested in this field, is there any ways you can make their dreams come true, like by offering internship to young Africa?
I would love to be able to have people in my lab. I take students from two grad programs. Sorry, I didn't catch the last part. I think the question was basically how can we get your people into our labs, and that. So unless I'm wrong, I'll answer that question.
Yeah. Yeah, exactly. That's the question. Like,
Yeah.
how can you get the young
Yep.
Africans in the
So if anyone is
lab?
interested, I would love to have you as a grad student. I take students from two. Sorry, go ahead. I think you're breaking up for me a little.
Training, yeah.
Okay. Anyway, I'll answer the question and we'll if I'm answering, if that doesn't answer it completely, shoot me an email. So if anyone wants to work on what my lab is working on, apply to grad school at the University of Minnesota. I can only take students who are in the program. So you basically have to apply to grad school, and then you can point as my lab as your target. And if you get in, I would love to work with you. And then most of the PIs in the Build-A-Cell are associated with grad programs. So if you're interested, you can always email any one of us and say you want to work with us, and we'll give you directions to the specific grad programs from which our students are coming from. So we're very happy to work with you. And if you want, please email. And for all of you that are not in the US, if you get into a US grad program, they will sponsor your visa. I was on a student visa as a grad student in the US too, and I know it's a very seamless process. The university will take care of your F visa, and they'll give you all the necessary paperwork. So you don't have to worry about immigration status or figuring that out on your own. If you get into grad school, your visa will be you'll basically get an invitation and an F visa. So that's not a problem.
Also, Kate, before we go on the other questions in the chat, I would like to inform everyone on this call that as SynBio4ALL Africa, we shall keep in close contact with Kate and the whole team at the Build-A-Cell and the whole team at University of Minnesota to ensure that for any opportunities which are open there, we can try to share with you all those opportunities. And if there is an opportunity with Kate, and maybe Kate has shared with us, we shall always avail those opportunities to you. And I'm sure those opportunities will come as time go on. And as SynBio4ALL Africa, we are very happy to always share those opportunities with you. We can go forward, Kate.
Yes.
Sounds great. Thanks again, Kate, for being here with us. If any of you are interested more in our course that we provided, I just posted a link of like what we have what we have in the last intro course last year, and we are working on the intermediate course. And like, please feel free to like stay connected with us. Kate also like gave a lecture in the class that were available online. So if you're interested in search how to grow almost anything, I think Kate gave one lecture about the cell-free system, and that could be like a good start on like how to learn further about the basics of these things. And I think like for the interest of time, we'll finish our webinar for today, and we'll stay in touch. Thanks a lot, everyone, for coming. And again, thanks, Kate.
Maybe Ice before we finish. I would like to appreciate Kate for fixing this time and being with the whole community from Africa. I would like to request everyone on this call, please, you can follow us on our Twitter, you can follow us on LinkedIn, and we have our email, our email, SynBio4ALL@gmail.com. Please follow us for any updates. We shall keep in close contact with Kate. If at all you need anything from Kate and you can't reach her, please don't fail to reach out to me. You can reach out to me on my personal email. I'm sharing it in the chat. I will be happy to share your question or your concern with Kate, and Kate will really respond to everyone. So for that matter, I'm very happy that we have had one of the people we have been looking at having on this platform, and we have had her. Please take this opportunity to continue engaging with her, to continue engaging with us. We shall always connect you to her, and more engagements will continue moving, and we look forward to collaborate more with Build for Cell and University of Minnesota. Thank you so much, Ait, for moderating this session. Thank you.
Thank you. Thank you all.
Thanks, everyone.
See you and have a great
Thank
great
you.
weekend.
Thank you very much, Lee. Nice lecture. All right then.
Have a great day.
Thank you.
Okay. Yeah.
Bye.
Recording.